Hey guys, welcome back to the channel. So in this video, we will be learning about general histology. General histology, the complete general histology of first year. So before I start this video, I just want to tell you that histology and embryology are the two histo and embryo are two units in anatomy where most of the students don't focus and actually this is where they don't score. Okay, so don't score. To be frank, if you to be frank, histo and embryo are the easiest subject if you give time to them. Around 1 week is enough to complete the entire histology and embryology. So if you give 1 week, you can complete the entire histo and embryo. Around 2 days is enough for 2 days is enough for histo, and then for embryo around 3 to 4 days. The entire syllabus, okay, for the entire syllabus. I'm talking about the final exams. I mean the university exams. For university exams, you require only 3 to 4 days for embryo and 2 days for histo. But that does not mean you have to read them in the last moments. Okay, so what most students do this mistake, that they keep histo and embryo pending, and then at the last it gets burdened. So finally your annat aggregate will come less. Okay, so let's start the video. Guys, in my video I have three major parts. I mean, in the presentation, the microscopic view of the slide, and then coming to this part, this is where you study about the key points about the slide, and the last one is the diagram. Okay, so the microscopic view is only useful during your practicals to identify the slide. In practicals, you might be asked to identify, I mean, the spotter, or it can also be a discussion topic. In spotter, you have to identify the slide. This, for example, this slide is placenta. You have to identify it, and you have to write two key points about them. Okay, so this is for spotter. For discussion topic, you have to draw this diagram, and then again you have to explain these things to your external examiner. This thing is again the key points. So I have kept in my mind during preparing of the presentation that this video should be helpful for both your practical exams and as well as your theory exams, theory as well as practical. So okay, so if you study through my video, you will be able to ace both your theory and practical exams. Okay, wait a minute, let me erase all this. Let's start with the topics. So in this video, I am dealing only with the general histology. The next video, I mean, in this series will be on the systemic histology. Go and have a check on that also. I think it will take you around 1 day to complete the entire histo, but I guess you need one more day to revise. You have to go through the video again and again, put 2x or 1.x, do whatever you like, do that. Make sure you remember all the key points, all the diagrams, everything. Okay, let's start with the video. The first slide, placenta. Coming to the slide, the microscopic view is somewhat like this. Have a look at it, microscopic view. if you see this type of image, I mean in the microscope, this is placenta. So basically this is containing chorionic villi sections. I mean the sections of chorionic villi are seen here. Okay, and then these villi are containing embryonic connective tissue. मतलब इधर देखो, या एक कोरियोनिक विलय हो गया और इसमें दिस इज एम्ब्रियोनिक कनेक्टिव टिश्यू. एम्ब्रियोनिक कनेक्टिव टिश्यू हो गया. नेक्स्ट, इट कंटेन्स फीटल कैपिलरीज and surrounded by outer syncytiotropoblast and inner cytotropoblast. So let's go deep into it. This region is the embryonic connective tissue. Done? Okay. Now this thing is the capillaries, fetal capillaries. Fetal capillaries. The cell's inner layer is the cytotropoblast. The outer layer is syncytiotropoblast. Okay, done. The same thing, see, villi with embryonic connective tissue core containing fetal capillaries and surrounded by outer syncytiotropoblast, inner cytotropoblast. Under cell-like layer, okay, cell-like layer. Outer is a uniform layer. It is merged. Okay. Cytotropoblast, syncytiotropoblast. That's all. Next point. Between the villus, we have maternal blood cells. What is this space? This space is called as intervillus space. Intervillus space. And it contains maternal blood. Okay, maternal blood. Done. Okay. So this finishes your placenta. The slide again. Points, key points. Sections of chorionic villi seen. Villi contains embryonic connective tissue, and it contains fetal capillaries, surrounded by outer syncytiotropoblast, inner cytotropoblast. Done. Intervillus space, maternal blood. Diagram, have a look at the diagram again. So the diagrams here, I have taken it from Balakrishnan Shetty. That book is having a horizontal layout. You can find it in your library, or you can even buy it from Amazon. I will provide the link below. Okay. Mesenchyme is nothing but embryonic connective tissue, intervillus space, fetal vessels, cytotropoblast, syncytiotropoblast, chorionic villi. Done. Next one, skin. Coming to skin, we have two types, thick and thin. Okay, so the main distinguished feature about thick and thin skin is that thick skin has thick keratin layer, and then thin skin has thin keratin layer. Okay, so you can find thick skin having a thick keratin layer, and then wait a minute, see, thin skin has thin keratin layer. Done. And then thin skin has hairs. Okay, hairs, but thick skin does not have hair. Done. इसको देखो, this is the keratin layer. You can identify the slide just by seeing the keratin layer that it is a thick skin, and thick skin does not have hairs. Okay, does not have hairs. Now coming to thin skin has hairs in it, but there is exception for this. Exception is what? Scalp. Scalp is actually a thick skin having hairs. That's all. Pura ho gaya. Now coming to the key points and the diagram. थिक स्किन में बाल नहीं होते, सो नो हेयर फॉलिकल्स. If there is a hair follicle, along with the hair follicle, there should be a sebaceous gland. See, there should be a sebaceous gland. This is the oil secreting gland for lubricating the hair. Okay, so there is no hair follicle, so obviously there will not be any gland along with it. That gland is called as a sebaceous gland. Okay, and then the epidermis, outer epidermis, is containing stratified squamous epithelium. Okay, stratified squamous epithelium. Since it is a thick skin, it is containing thick keratin layer. So I'll repeat it again. Oh my god, wait. Thick skin does not have hair. If there is no hair follicle, there is no sebaceous gland. Okay, and then outer epidermis, stratified means layer. Squamous means the squamous cells, the outermost surface like this. Okay, squamous cells, flat cells with thick keratin layer. Okay, that's all. Inner dermis contains sweat glands. So you have to remember that the entire body is having the capacity to sweat. Okay, sweating is possible through entire body in human beings. In humans, sweating is possible through the entire body surface. So either it is thick skin or thin skin. Both these skin have sweat glands, pasina bahane ke liye. Okay, sweat glands in their dermis layer. So in the dermis layer, dermis not epidermis. Okay, dermis, remember again. So both the thick skin and thin skin has capacity to sweat. You just understand the concepts. Okay, the entire body is having the capacity to sweat, but the entire body is not having the capacity to secrete oil. So that's why you get dry skin. Okay, understood? That's all. But pasina body का सब parts में पसीना आता है. Then coming to the epidermis, 5 layers we have. Okay, the outermost layer is corneum. Corneum means keratin layer. The last layer is base or basal layer. Done. The middle layer is granulosum layer. Granules contains granules. See, I'll tell you again. First remember the 3: outer, inner, and then middle. These 2 you have to remember. Okay, see, the outermost is corneum. It contains thick keratin layer. After that, it has connective tissue. Okay, connective tissue layer. This is called as stratum lucidum. This somewhat looks like a space. Okay, we have to draw like a space after first layer in your diagram. Then the third layer is granulosa layer. So the name itself suggests that it contains granules. See, it has granules along with the nucleus. Granules are there, right? Then the last layer is basal. So it is the base. It is occupying the base layer of epidermis. Done. It is columnar in shape. Columnar. And then spinosum is the fourth layer. Spinosum is polyhedral in shape. जैसे भी मन करेगा वैसे कर दो. Okay, done. We have done the epidermis now. So this much is your epidermis. Let me repeat the layers again. The first layer, corneum. Done. The middle layer, granulosum. The base layer, basal. Here this is a connective tissue layer. It is, you have to draw like a gap after corneum and between the granulosum. This layer is called lucidum, stratum corneum, stratum lucidum, stratum granulosum, and then stratum spinosum. This is polyhedral. Done. So we are completed with epidermis. Now coming to the dermis. As I told you, dermis contains sweat glands. That's all you have to remember in the dermis. So write like this: normal cells. So this is a sweat gland. So write many sweat glands. Okay. So invagination from epidermis into dermis is called epidermal papilla. Invagination from dermis into epidermis is called as dermal papilla. That's all. We are completed with thick skin now. Coming to thin skin, this is very easy to identify. We have hair follicle in it. That's all. Hair follicles. So identification is done. For identifying thick skin, the corneum is enough. Corneum, stratum corneum, or the thick keratin layer. Done. For identifying thin skin, hair follicle is enough. Now, again coming to the key points. Outer epidermis. This, see, for skin, we have to remember stratified squamous layer. Okay, stratified means stacked layers. Okay, stacked layers. Stratified. Squamous is the flattened cells with thin keratin here. Here it is thin keratin. Okay, see, the keratin layer is very thin here. Wait a minute, we'll see the thick. See, this is thick keratin, whereas this is thin keratin layer. Okay, we have only 3 layers in thin skin. Thin skin में only 3 layers होता है. 3 layers only in thin skin, whereas in thick skin 5 layers. Let's repeat the 5 layers again. First one, corneum, lucidum, granulosum, spinosum, and basal. Right. So in the thin skin we have first layer, fourth layer, and fifth layer. What are those? Corneum. Wait a minute. Sorry. Spinosum and basal. Right. 1, 4, 5. Done. Very easy, right? See. This is corneum. Again, and this is spinosum, corneum. This layer is called as basal. Done. See, they have told you keratin layer, and then they have mentioned epidermis. They have not mentioned the layers here. They've just given, I have written the layer's name. You can write the layer's name also here. Invagination of epidermis into dermis is epidermal papilla, and then dermal papilla is invagination of dermis into epidermis. Done. What is this? This is now a hair follicle. Hair follicle is this thing. This thing is a hair follicle. Okay, that's what they have mentioned here. And now this is the gland for the hair follicle. Lubrication, oily secretion. Okay, this gland name is called as sebaceous gland. The hair follicle has the muscles for it, smooth muscles. This is called as erector pili muscles, this layer. So if you observe, these muscles are smooth muscles. See, smooth muscles like this, like this, like this. Okay, smooth muscles. Okay, the name for this muscles is erector pili. So what do they help in piloerection? Goosebumps, piloerection. Okay, done. Coming to the dermis now. We are completed with epidermis. Now dermis, again only sweat glands are present. See, sweat glands. Okay. Outer layer done. Squamous, thin keratin layer. Stratified squamous, thin keratin layer. Inner dermis contains hair follicle, sebaceous gland, erector pili, sweat gland. See, it is containing, dermis is containing hair follicle, sebaceous gland, erector pili, and then sweat glands. Done. Very easy, right? Okay, then coming to the next slide. The next slide is a large artery. So coming to arteries and veins, large artery is having a slide like this. See, it has more dense pink color, whereas your large vein is having a cross section again similar to that, but it is not so darker in color. See, this is large artery, this is large vein. Okay, so large artery, large vein, large artery, large vein. Okay, now coming to medium sized artery and large artery. The large artery is like this, whereas the medium sized artery is like this. One easy identification point is that the large artery, we cannot see the entire artery through the microscope. I mean, the entire circular structure we are not able to see in the case of large artery, because it will be like this in the microscope. Okay, we can see only this field of the large artery, whereas in the case of medium sized artery, the whole artery lumen is seen. The lumen is seen completely, whereas in large size it is not possible to see the complete lumen. Not possible. Okay, then coming to the key points. 3 layers: intima, media, externa, also called as adventitia. Intima, media, externa. Inner layer intima, middle layer media, outer layer externa or adventitia. So the middle layer is thickest, both in large size artery, as well as medium sized artery. The middle layer is thickest. Okay, thickest. But in the case of large artery, elastic fibers are more compared to the smooth muscles. Why? If you remember from physiology, the large arteries are also called as Wind-Kessel vessels. Wind-Kessel. Do you know about the Wind-Kessel effect? Wind-Kessel. Okay, Wind-Kessel. So for Wind-Kessel effect to be in action, there should be enough elasticity for the artery. So in order to provide that elasticity, elastic fibers are present. Elastic fibers. So in the media, thick elastic fibers are present compared to smooth muscles. More elastic fibers, less smooth muscles. Coming to the last point, we have vasa vasorum present in tunica adventitia. So adventitia is the last layer, right? The outermost layer, tunica externa or tunica adventitia. In this layer, we have vasa vasorum. What is vasa vasorum? These are blood vessels that supply the large blood vessels. Blood vessels supply large blood vessels. So here, these blood vessels supply the large blood vessel. So here the large blood vessel itself is the artery. So this blood vessel is supplying the blood to this artery. Okay, location aorta, common carotid. That's all. So yes, 3 layers. Media is thickest, elastic layer. I mean, the more elastic fibers are present, less smooth muscle fibers, and then vasa vasorum present in tunica adventitia. Location aorta, common carotid artery. That's all. Finished with large artery. Now coming to medium sized artery. You can identify it by complete lumen. 3 layers again, intima, media, externa. Media is again the thickest, but here smooth muscle fibers are more. Okay. Now I did not tell you about this thing, right? I did not explain you about the diagram. So the first thing you have to write is the endothelium. These are squamous flat cells, endothelium. And then we have to write a connective tissue layer. This connective tissue layer is called as subendothelial connective tissue. You can see here, subendothelial connective tissue. Okay, so this region is subendothelial connective tissue. This region. Next, we have to write a wavy lamina or sheet-like structure. This is called as internal elastic lamina, and then we have more elastic fibers like this, and then few smooth muscles. So most of your large artery is made up of more number of elastic fibers. So see, comparatively there is more elastic fibers compared to smooth fibers. That's all. Elastic fibers more compared to smooth, so it is less. Now again similarly we have another limiting, I mean the external elastic lamina. Okay, lastly we have adventitia containing vasa vasorum, external elastic lamina. So in the medium sized artery we have prominent internal elastic lamina. So we saw elastic laminas, no? Here this is the internal elastic lamina. This thing in medium sized artery is prominent. That's all they are saying. Then smooth muscles are more compared to large sized artery. Okay, we know. And then here remember vasa vasorum is absent. In the case of large arteries, vasa vasorums are present. Vasa vasorum is it's an adaptation for blood supply for the blood vessels itself. So only in the case of large arteries vasa vasorums are present. In case of medium sized and small sized arteries we don't have vasa vasorum. Example for medium sized artery is radial हाथ में दो, yeah, like this, biceps here, radial artery, okay, radial artery, ulnar, okay, ulnar artery. And then in the leg we have popliteal artery. This is the popliteal fossa, this popliteal artery. Okay, that's all. Radial, ulnar, popliteal. So for large artery it was aorta, common, carotid, right? In this case radial, ulnar, popliteal. Finished. Now coming to diagram, we have again the same endothelium and then subendothelial connective tissue, subendothelial connective tissue layer. Wait a minute. Yeah. Oh my god. subendothelial connective tissue. This thing is subendothelial connective tissue layer. I'm sorry, I'm new with this iPad. I'm getting adjusted to it. Okay, and then we have prominent internal elastic lamina. The next layer is the tunica media, which is again the thickest. But in this medium-sized artery, smooth muscles are more compared to elastic fibers. Right? Fibers. Done. Again, external elastic lamina. This is tunica adventitia or tunica externa. This does not have vasa vasorum. That's all done. Completed with external elastic. Completed with the medium-sized artery. Coming to large vein, it is having 3 layers again: interna, media, adventitia. Okay, so in this case, see, in large-sized artery and medium-sized artery, which was the thickest layer? That was media, right? The middle layer, media, was thickest. But in the case of large vein, we have adventitia layer as the thickest. Adventitia layer as the thickest layer. See, you know how to identify the slide, right? I told, for the case of large artery, it is more darkly stained. For the case of large vein, it is lightly stained. Light stain. Dark stain. See, you can see it, no? Large artery, large vein. See, done. Now these 3 layers are done. Tunica adventitia or externa, thickest with smooth muscles. Okay. Tunica adventitia, we'll see the diagram. Tunica intima, this much. Subendothelial, endothelium, subendothelial connective tissue. The next layer is tunica media, tunica adventitia. Done. The first layer is tunica intima, second layer is tunica media or the middle layer. But in the case of large vein, we have adventitia layer as the thickest layer. So you have to write this layer larger compared to your media. Okay, so less and more. What did we study here? Tunica adventitia contains longitudinal smooth muscles. Tunica media contains circular smooth muscles. So see here, inner circular, whereas the outermost layer, the tunica adventitia, contains longitudinal. See, these are longitudinal fibers. I mean, in the cross section, the longitudinal fibers looks like this, whereas in the cross section, the circular layer looks like this. That's all. Tunica media and tunica adventitia. Location, coming to the location, we have it in IVC and SVC. So heart, superior vena cava, inferior, IVC. Done. That's all from large vein. Next, coming to the cartilages, we have 2 slides: hyaline cartilage and elastic. We have 3 slides: hyaline, elastic and white fibro. Okay, so see, if at all you get confused, you will get confused with hyaline and elastic cartilage. So see the slides before. See, this is dark stained, right? Dark stain is hyaline. Okay, hyaline cartilage, whereas the light stain is elastic cartilage. And the next distinguishing feature is that it contains aggregate like cell nest, okay, see, in clumps. These are called as cell nest. Chondrocytes are present in this cell nest, whereas in the elastic cartilages, it is not in the clumps. They are not present in cell nest. They are, if at all they are present, they are very rare, okay, compared to hyaline cartilage. We are done with microscopic view now. Okay, so perichondrium is the outermost layer of cartilage. Okay, see, in the case of bone, it is periosteum. In the case of cartilage, it is perichondrium. So perichondrium is the outermost layer. In that layer also we have two layers again. So two layers. The outermost is fibrous. Okay, the inner is cellular. Let's see it. See, the outer. So this until here is the perichondrium. In the chondrium, in the case of cartilage, whereas in the case of bone, it is periosteum. Okay, osteum. This okay, until here is your perichondrium. Perichondrium again has two layers: fibrous, outer, inner cellular. Okay, so this is fibrous layer, outer fibrous layer, and this is inner cellular layer. See the cells here. This is fibers, total fibers. Done. And then what did I tell you? Chondrocytes are present in cell nests. See, these are nothing but in the clumps, right? Clumps are also called as cell nests. You don't have to tell clumps, okay? You have to tell the scientific term cell nest. Chondrocytes. So these are chondrocytes. Present in cell nests. I mean in an aggregate manner. Next, the territorial matrix is surrounding cell nest. Interterritorial matrix between two cell nest. Okay, it's very easy. See, this purplish color is there, no? This is nothing but the territorial matrix. Whereas this lightly stained region is interterritorial matrix. So darkly stained territorial matrix is newly produced or fresh, whereas the light stained interterritorial matrix is old one. Okay, that's all. Location, again coming to the location of hyaline cartilage. It is present in ribs. I mean the coastal cartilage, and as well as articular cartilage. So imagine this as the bone. We have cartilages here. This is the capsule of the bone, I mean the joint. This cartilage is made up of hyaline cartilage. Okay, done. Coming to next slide. This is the elastic cartilage. We know how to identify the slide. Light stained elastic cartilage, and then this does not have cell nests absent. Okay, so again, outer fibrous perichondrium, inner cellular perichondrium. Matrix contains elastic fibers. See, the name itself tells you elastic. So this is the peculiar feature of special feature of elastic cartilage, right? Elastic cartilage should be elastic. So the matrix contains elastic fibers. Okay, matrix contains elastic fibers, and then there are numerous chondrocytes, but they are not present in cell nest. They are present everywhere, but few nest cells. That's what I told you. See, there are numerous chondrocytes. Of course, chondrocytes are the cells of cartilage. Okay, so the principal cells of cartilage are chondrocytes. They have to be present everywhere. They are present everywhere, not in the cell nests. That's all. Then location, external ear and epiglottis. See, you have to remember triple E, like the film Triple R. We are remembering it triple E. What? Elastic cartilage is present in external ear and epiglottis. External ear. Elastic cartilage is present in external ear and epiglottis. That's all. Coming to the perichondrium, we have outer fibrous, inner cellular cells, fibers. Coming to matrix, we have the elastic fibers throughout the matrix, elastic fibers. Now, chondrocytes are present everywhere. Look at it. They are present everywhere, right? These are chondrocytes, no? They are present everywhere. Almost the entire region is present with chondrocytes, but they are not present as cell nest. See, where is the cell nest here? We did not observe one cell nest also. They are in rows, I know, but they are not present like this. If it is like this, it will be a cell nest. So there is no cell nest, very few cell nests. What else did I miss? I think everything is covered. Okay. Next, coming to white fibrocartilage. White fibrocartilage, I'll tell you the example where it is present. It is present in intervertebral disc. Okay. We'll see. Yeah, intervertebral disc is the location. That you can identify it very easily. This is the easiest slide that one can identify white fibrocartilage. Okay. See. Hyaline cartilage, elastic cartilage, hyaline, elastic, white fibro. Three are also very easy, right? Elastic is lightly stained with no cell nests, whereas highly stained is hyaline cartilage with cell nests. That's all. This is white fibrocartilage. This is somewhat like this. Just observe this. Very easy slide. The peculiar feature about this is it does not have perichondrium. It is not covered by perichondrium, and it contains abundant collagen fibers. Collagen. Whereas in the elastic cartilage, it was elastic fibers. Here it is collagen fibers. Collagen fibers. Okay. And then scanty chondrocytes in rows. Scanty chondrocytes. So chondrocytes are very less compared to the amount of collagen that is present. See, collagen, there is too much of collagen in here. These are collagen fibers, whereas chondrocytes are very less, and if at all they are present in rows. Bundles of collagen, chondrocytes in rows. Next, we completed cartilage also. Now coming to the slides of bones. We have TS and LS of bone. TS means transverse section and LS means longitudinal section. So if you get the slide of bone during your practicals or theory exams, just think that it is a God-given gift, okay? Because TS and LS of bone is the easiest slides that anyone can get, okay? Coming to TS of bone. This is how TS of bones slide is, okay? Transverse section. This is a transverse section. Now coming to LS, this is how longitudinal sections are present of bone. Done. Transverse, LS or longitudinal. TS of bone. Now periosteum is present. Okay, periosteum, again as I told, periosteum is present, outer fibrous, inner cellular, same. Now we have lacunae containing osteocytes. What are osteocytes? Osteocytes are the bone forming cells, okay, the principal cells of bone. Principal cells of bone are osteocytes. So where are they present? They are present in the spaces called as lacunae. So lacunae is a space, empty space, okay. Lacunae is an empty space. Now coming to lamellae is a sheet like things where the lacunae are present. Lacunae is nothing but the space. Now coming to the lamellae. So what is lamellae? Lamellae is a sheet like structure, okay. So these lamellae are present between the lacunae. So this is a lacunae, this is a lacunae, this is a lacunae, right? So similarly on the other side these will be the lacunae. So now this region is considered, this region is considered as the lamellae. See, lamellae. So among the lamellae we have three types of lamellae. Which are the three? The circumferential, interstitial and concentric. See, three type of lamellae. Which are those? Circumferential, concentric, interstitial. Circumferential is the lamellae that is present on the circumference of the bone. See, this circumference of the bone. Circumference of the bone. This is the circumference of the bone, the outer end part, right? So this region is going to be the circumferential lamellae. Now coming to interstitial lamellae, this region is the interstitial lamellae, whereas the concentric lamellae is these regions. Circumferential, concentric and interstitial. Very easy, right? Yeah. And this thing is the Haversian canal, and one complete unit is called as the Haversian system. There are connecting channels between two Haversian systems, and that is called as the Oaksman's canal. 3 points. Which are those? Haversian canal. The complete unit is called as the Haversian system, and the connecting channels between two Haversian systems or the Haversian canals is called as the Oaksman's canal. Done. Very easy. Very easy. We'll see the labelings now. As I said, the osteocytes in lacunae. Osteocytes in the lacunae. So this is a lacunae, this is an osteocyte. This is a lacunae, this is an osteocyte. You have to write like this, okay? Don't forget to write like this. Then the next one, interstitial lamellae, Haversian canal, concentric lamellae, Oaksman's canal, circumferential lamellae. Coming to periosteum. Periosteum is again divided into outer fibrous, inner cellular. See, the outer fibrous and then inner cellular. Outer fibrous, inner cellular. Very easy, right? Very easy. Outer fibrous and inner cellular. These are cells, cellular. We completed with transverse sections of bone. Now coming to LS of bone, longitudinal section of bone. It is somewhat like this. Longitudinal sections of Haversian system containing central Haversian canal. Right? This is a Haversian canal and one complete unit will be the Haversian system. Haversian system. This is what we saw in this. One complete unit is the Haversian system. Here, that one complete unit is this thing until here. Haversian system. Haversian canal. Interconnecting thing. This thing. Wait. This thing that connects two Haversian canal or two Haversian system is called the Oaksman canal. See, where is Oaksman canal? This is the Oaksman canal. Done. Haversian canal. Now again, osteocytes in the lacunae. Now this middle region is the lamellae. Very easy, right? Yeah. Oaksman canal interconnecting Haversian canals. Osteocytes in lacunae arranged in lamellae. So osteocytes in lacunae arranged between the lamellae. Osteocytes in lacunae arranged between lamellae. These are lamellae. Osteocytes are arranged between this thing. Skeletal muscles. Skeletal muscle and cardiac muscle, you can identify it very easily. See, coming to skeletal muscle and cardiac muscle, this is a pretty. Actually, most people don't know how to differentiate between skeletal muscle and cardiac muscle in the case of microscopic view. See, skeletal muscle has large space between it, whereas see the space, it's so tiny spaces here. Skeletal muscle has large spaces, whereas it's densely packed. Cardiac muscle is dense packed, whereas skeletal muscle, it's not so dense packed. Done. Very easy. Again, coming to the points, peripheral, multiple flat nuclei. See, this is a skeletal muscle fiber or a single cell. Okay, we consider this as a single cell in the case of skeletal muscle, and now it has multiple flat nucleus along the periphery. It is unbranched, long, cylindrical fiber. It's the same thing I told you. This is not branched, it is single all over the course, and it is long, cylindrical fiber. Dark and light bands are seen. See, dark, light and dark, light, dark, light, dark. Okay, dark and light bands are seen. That's all. Muscle fiber, dark and light bands, peripheral flat nucleus. Sarcolemma is the plasma membrane of the cell or the fiber. Coming to cardiac muscle, as I told you, it is densely packed compared to your skeletal muscle. Coming to the key points, short cylindrical fibers. So these are short cylindrical fibers, whereas the skeletal muscle was unbranched, long cylindrical fiber, whereas cardiac is short cylindrical fiber, and it is branched. Okay, it contains single central oval nuclei. Muscle fibers are branched. Intercalated discs are seen and are made up of desmosomes, gap junctions and tight junctions. Okay, so desmosomes, gap and tight. Three type of junctions are there: desmosomes, gap junctions, tight junctions. for impulse transmission. Remember the function, impulse transmission of impulse. So thus it acts like a syncytium. Okay, the special feature of cardiac muscle. Short cylindrical muscle fiber containing central oval nucleus, contains intercalated discs made up of gap junction, tight junction, and desmosomes, right? And they are branched. That's all, I think. Yeah, short cylindrical central oval muscle fiber. Yeah, that's all. Dark and light bands are seen again. Branching pattern, intercalated disc, single central oval nucleus, sarcolemma. Done. Okay, guys. So coming to the ganglions, we have two ganglions now. So sympathetic ganglion and the spinal ganglion. The spinal ganglion is also called as the dorsal root ganglion. See, sympathetic ganglion location is sympathetic chain. So if you know, sympathetic chain lies beside the vertebral column. So imagine this as your vertebral column, the bones of vertebral column, and then here is what the sympathetic chain. Okay, so in this we have ganglions called as the sympathetic ganglion. Okay, so this is how a sympathetic ganglion looks like. It is pale, I mean it is not densely stained, not dense stain. Okay, stain. Whereas your spinal ganglion, see, it is densely stained. See, you can observe, right, which is dense, which is not dense. See, the dense stain, whereas this is not dense stain. Okay, so spinal ganglion. Coming to spinal ganglion, where is it present? Spinal ganglion is present, as the name suggests, spinal ganglion. This is the spinal cord. This is the spinal nerves, the dorsal and ventral. Okay, so this will be your dorsal root and this will be your ventral root in this. Okay, the dorsal root, ventral root, right? This is dorsal spinal nerve, ventral spinal nerve. So from here we get a ganglion here. If you remember, from the dorsal spinal nerve we have a ganglion which is a pseudo unipolar ganglion. Okay, so this ganglion is called as dorsal root ganglion. Dorsal root ganglion or also called as spinal ganglion. Done. Okay, so coming to key points, we have in sympathetic chain, it is covered by thin connective tissue capsule. Whereas in the spinal root, I mean the spinal ganglion or dorsal root ganglion, it is thick connective tissue capsule. The capsule in spinal ganglion or dorsal root ganglion is thick, whereas in sympathetic ganglion the capsule is thick. Okay, here ganglionic cells are arranged in groups between nerve fiber bundles. So ganglion cells are arranged in bundles. Okay, see this, ganglion cells are arranged in groups. They are arranged in groups between the nerve fiber bundles. These are nothing but nerve fiber bundles. Okay, So ganglionic cells are arranged in groups between nerve fiber bundles. Okay. The last thing that these neurons are pseudo unipolar neurons. As I told you, this is a pseudo unipolar neuron, right? So this is a pseudo unipolar neuron. It contains large pseudo unipolar neuron. Here we have the nucleus at the center, central nucleus surrounded by satellite cells. So, okay, the central nucleus surrounded by satellite cells. These are satellite cells. So, the function of satellite cells is nourishment and what is that? Nourishment and insulation. See, the function of satellite cells is nourishment and insulation. Okay, very easy. Location is sympathetic chain for sympathetic ganglion. In the case of sympathetic ganglion, we have cells like this, and the nucleus is not at the center. Small multipolar neurons with eccentric nucleus, not at the center, eccentric, okay, surrounded by satellite cells again. In this case, we have small multipolar neurons, whereas in dorsal root ganglion, the neurons were pseudo unipolar. In the case of sympathetic, it is multipolar. Okay, small multipolar, there large pseudo unipolar. Go through the slide once. See, this is not densely stained, and you can clearly see the ganglionic cells in bundles. In the case of what? In the case of dorsal root ganglion. Whereas in sympathetic ganglion, they are not arranged in bundles. Okay, scattered ganglionic cells between nerve fibers. So these are nerve fibers. The ganglionic cells are scattered everywhere with eccentric nucleus and satellite cells. Very easy, yeah. And then we have a capsule, thin capsule in the case of sympathetic. Scattered ganglionic cells, eccentric nucleus, satellite cells, nerve fibers. Easy, yeah. Done. Spinal ganglion was also done. Now coming to the lymphatic system, we have 4 slides from lymphatic system. The lymph node, tonsil, then thymus, and then spleen. Okay, so lymph node, tonsil, thymus, and spleen. These are 4 slides from the lymphatic system. Coming to lymph node, this is how the microscopic view is. We have a capsule. We have a space called as the subcapsular space. So this is a capsule, this is subcapsular space. So sub means below the capsule. Below the capsule we have subcapsular space. Okay, the invaginations of the capsule is called as trabeculae. Okay, so between the trabeculae we have lymphoid follicles, and the center portion is called center portion of the lymphoid follicles is called germinal center. Capsule, subcapsular space. Wait a minute, I'll rub all this. Okay, capsule, subcapsular space, trabeculae, and then lymphoid follicle, germinal center, right? Okay, if you observe the key identification point for lymphatic, the lymph node is this thing. We have trabeculae and lymphoid follicles. Okay, trabeculae, lymphoid follicles. That's all. It is pretty easy, but most of the people get confused while identifying in the practicals. Don't get confused. Go to your lab, see the slides again and again, see this video. Okay, so fibrous capsule and subcapsular space, as I told. Now imagine your lymph node as a circular ball. Okay, this is a circular ball. Okay, and in the middle we have a medulla, outer cortex. So this region, this region is the cortex, this is the medulla, right? We'll rub all this. Okay, so cortex and medulla. So this region between the cortex and medulla is called as paracortex. Paracortex. Okay. Outer cortex and inner medulla differentiated. Okay. Then we have a germinal center. We have lymphoid follicles. In the germinal center, we have B lymphocytes. In paracortex, we have T lymphocytes. In medullary cords or macrophages. Okay. See, we studied until cortex. Now, coming to medulla, we have cell-like these cells, no? This region is called as, these are called as cords, medullary cords. And this part is called as medullary sinus. Medullary sinus. The medullary cords and the medullary sinus. Let's see the labelings. See, this is medullary cord and this is medullary sinus. Now, this is trabeculae. This is germinal center, lymphoid follicle, subcapsular space, the capsule. Okay. And then what they have told us, in the germinal center, in this region, we have B lymphocytes. B lymphocytes. In the paracortex, as I said, the region between medulla and the cortex, we have T lymphocytes. T lymphocytes. Okay, T lymphocytes. And in the medullary cords region, we have plasma cells and macrophages. Okay, so let's revise again. Capsule, subcapsular space, lymphoid follicle, germinal center, paracortex. This is cortex, this is medulla, and then sinus, this is medullary cords, right? In the germinal center, we have B lymphocytes. In the paracortex, we have T lymphocytes. In the case of medullary cords, we have plasma cells and macrophages. Plasma cells and macrophages. See, plasma cells and macrophages. Okay, this slide is also completed. Coming to tonsil. So most of you will get confused with lymph node and tonsil. So how to identify or how to differentiate between a lymph node and a tonsil is very easy, guys. You have to focus on this part. See, this part. This type of structures is not visible in the case of lymph node. In the case of lymph node, there is no mucus acini. So these parts are nothing but mucus acini. Okay. So coming to the epithelium, it is again similar to skin, but this tonsil does not have keratin layer. there is no pigmentation. Okay, so similar to skin, nothing but as stratified squamous. Okay, but it does not have keratin or pigmentation, so non-keratinized. Stratified squamous non-keratinized epithelium, right? Easy. Now coming to the shape of tonsil, it is somewhat like this, and these regions are called as crypts. The tonsillar crypts. So tonsillar crypts are depressions. Okay, so along the depressions, along the crypts, we have lymphoid follicles. See, along the crypt, we have lymphoid follicles. Okay, and the epithelium is similar to skin, stratified, squamous, but here there is no pigmentation. So, non-keratin, okay, stratified squamous, non-keratinized, crypts, lymphoid follicle, germinal center, and then the main distinguishing feature is mucus acini. You know how to draw mucus acini? Mucus acini is you have to draw like this. Okay, the lumen should be big enough. Okay, lumen should be big, and the nucleus should be at the base, and it is a flat nucleus. Okay, it is secreting mucus secretion, mucus acini. Okay, we completed lymph node and tonsil. Lymph node, tonsil. See, this also has lymphoid follicles. This also has lymphoid follicles, but this does not has mucus acini. There is no mucus acini here, and there are presence of trabeculae here. There is no trabeculae. See, where is trabeculae? No trabeculae. That's how easy it is to differentiate between between a tonsil and a lymph node. Now coming to the thymus, this is how the microscopic structure is of thymus. Okay, so mainly it contains lobules. Okay, lobules, which is separated by incomplete septa. So lobules are present. You can see the lobules, right? See the lobules. So these are separated by incomplete fibrous septa. This is a very important point. Incomplete fibrous septa. So each lobule contains, so here we consider a lobule as a unit, principal unit. Okay, so if you consider this diagram, so if you consider this diagram, in this is a principal unit, also called as the lobule. It contains the cortex and medulla inside. See the peripheral cortex and central medulla. Here the peripheral cortex is densely packed and contains small lymphocytes. Whereas the central medulla contains large lymphocytes and contains the Hessals corpuscles. HASSA, Hills corpuscles. Okay, see, Hessals corpuscles. These are Hessals corpuscles. Large lymphocytes, small lymphocytes, cortex, medulla, Hessals corpuscles. Incomplete fibrous septa. So this is what incomplete fibrous septa. That's all. Very easy. Thymus. Lobules are present. Incomplete fibrous septa. Lobule contains a peripheral cortex. Small lymphocytes are present. And then central medulla, large lymphocytes, and Hessels corpuscles are present. Hessels corpuscles. The very important point is Hessels corpuscles. You don't have to forget. You should never forget Hessels corpuscles. Okay. Coming to spleen. So spleen, there is no point in getting confused, because when you see the slide of a spleen, you can see the red pulp very clearly, reddish slide. Okay, see, the tonsil is not so darkly stained. Even the lymph node is not darkly stained, whereas spleen, you can see the reddish texture here, reddish. Okay, so it contains red pulp and white pulp. Okay, so as soon as you see the reddish slide, this is a spleen. You just have to see this. The spleen contains a reddish texture. Okay, whereas the other slides, see this, does not have the reddish thymus does not have the reddish texture, whereas the tonsil is also not having the reddish texture. See the spleen, see tonsil has this thing, the mucus acini, whereas this does not contain mucus acini. Okay, whereas lymph node has trabeculae, but spleen, the major feature is red pulp and white pulp. So red pulp, you can just by seeing this thing, you can identify that this is a slide of spleen. Okay, it contains red and white pulp. Red pulp. See the diagram. We have a capsule. Firstly, we have a capsule of spleen, and then you have to draw like this. Okay, you have to draw like this, a spindle shaped sinuses, and then between these sinuses we have cords. So this region which contains cords and sinuses is called as the red pulp. Okay, and the rest of the region which contains lymphoid aggregate, this region is called the white pulp. See, white pulp contains lymphoid aggregate with eccentric central artery. Here again we have a artery which is eccentric in position, eccentric central artery. Okay, we're done with spleen. Again, contains red and white pulp. You can see the microscopic structure. Firstly, lymph node contains trabeculae, capsule, lymphoid follicles. Okay, tonsil contains, the main distinguishing feature is mucus acini. Okay, no trabeculae, and then in thymus we have lobules. Okay, in spleen we have no trabeculae, and then we have red pulp. That means we have a reddish texture of the slide, red pulp and white pulp. Red pulp contains splenic cords surrounded by sinusoids. Okay, white pulp contains lymphoid aggregate with eccentric central artery. Okay, done. Very easy. Capsule, white pulp, germinal center, eccentric placed central artery, splenic cords, sinusoids, which gives rise to red pulp. Okay. Coming to optic nerve. Optic nerve, you know the eyeball, right? This is the eyeball, and the nerve that connects the eyeball like this is the optic nerve. Okay, this is a cross section of optic nerve. You can easily see. You might get confused slightly with the ganglions. Okay, the slides of ganglions, but there is no point of getting confused, because in the case of optic nerve, we have two vessels. Okay, so the name of these vessels is central artery and central vein of retina. So see, this will be the region of retina, right? So it connects the retina, so it is called as central vein and central artery of retina. Okay, done. So myelinated axons are grouped as bundles. In the center we have central artery and central vein of retina, and it contains three coverings from inner to outer. Let us remember it as pad. Okay, अंदर से बाहर, pia matter, arachnoid matter, and dura matter. Okay, so pad from inner to outer, arachnoid matter, pia matter, arachnoid matter, and dura matter. Pia, arachnoid, and dura. See, pia, arachnoid, and dura. Okay, so and then we have myelinated axons grouped as bundles. Okay, myelinated axons grouped as bundles, and central vein of retina and the central artery of retina. Central vein and central artery, bundles of axons, myelinated. Okay, we are done with optic nerve. The next slide is about there are three slides of salivary glands. Okay, so coming to salivary glands, we have three: serous, mucus, and then mixed. Okay, the first one is serous. It contains watery secretion. Mucus is mucus secretion, and then mixed is intermediate. So serous secretion. This is a slide. Okay, wait. This is the slide of serous salivary glands, and then mucous salivary glands. This is mixed salivary gland. Okay, serous, mucus, and then mixed. Okay, I know you won't understand it right now. Okay, so let us discuss about the location. The serous salivary gland location is parotid. Okay, and then mucus location is sublingual. Okay, and then mixed is submandibular. Okay, so imagine a jaw of a human being. Okay, this is a parotid gland, the first one, and then below the tongue, second one, then below the mandible, submandibular. So we are coming from top to down: parotid, sublingual, submandibular, serous, mucus, mixed. Okay, and the acini that was present in what? In the tonsil was mucus acini. If you remember, these acini are mucus acini, right? Okay, serous is parotid, mucus is sublingual, mixed is submandibular. We're done with the examples. Now see, location: parotid only seen in serous acini. In the case of mixed, in the case of mucus, it is sublingual. In the case of mixed, it is submandibular. Serous acini, you can see, you can identify it very clearly. How serous acini, it is like somewhat like this. Okay, now serous acini contains biphasic staining. Wherever serous acini comes, you have to remember this word: biphasic staining. Okay, so coming to the structure of serous acini, we have pyramidal cells and small lumen. The lumen is small. Okay, in the case of serous acini. And then why is it biphasic staining? Because of why is it zymogen granules? Why is it biphasic stained? Because of zymogen granules. Look at this. This is a pyramidal shaped cells. Okay, pyramidal shaped, and then we have round basal nucleus, zymogen granules. You can see the zymogen granules. Okay, this is what uh, looks. This is how a serous acini looks like. Okay, done. This is a serous acini. Serous acini with biphase stain of pyramidal cells with narrow lumen. Okay. Coming to this part, this thing that you can see here is the connective tissue septa. Okay. Where is the labeling? Yeah. Connective tissue septa. And then intralobular ducts are seen, and then interlobular ducts are classified again into striated and intercalated. So coming to the duct system, we have two types: duct, intra and intra intralobular and interlobular, right? Intralobular, so within the lobule we have, so we consider this part and this part as a lobule here. So in this, whatever the ducts come is called as the intralobular ducts, which are again classified into striated and intercalated. Okay, so and then the rest is interlobular. So between the lobules, this is the region or the connective tissue septa, right? In this region, we have a ducts called as interlobular ducts. Okay. For striated duct, we have to write striations in the base, and we have to write the nucleus like this. And then next is intercalated. Where is intercalated? I don't see any intercalated. Yeah, intercalated duct. See, this is a intercalated duct. Normal. Okay. So let's discuss it again. We see interlobular, and intralobular. So intralobular are again classified into two types: striated and intercalated. Okay. So intralobular, within the lobules. There are two types of ducts: striated, which contains striations, and then intercalated. This is intercalated. Interlobular ducts are present in the connective tissue septa. Interlobular. Okay. Cuboidal cells. Done. The next slide is the mucous salivary glands. So you can easily identify serous salivary glands. So if at all there comes a confusion, it comes between the mucous and mixed salivary glands. So you can see the mucous salivary gland being darkly stained and is uniform, whereas the mixed salivary glands, you can easily see the serous acini also in the case of mixed. And then we have this things that is mucous acini. Okay. See, in the case of mucous salivary glands, we don't see much of that serous type. Okay, we only see mucus. See, this is mixed, this is mucus. Okay, so large mucous acini with flat basal nucleus and large lumen. Okay, as I said, flat basal nucleus, large lumen. Again, regarding the ducts, interlobular ducts, intralobular ducts, classified again into striated, intercalated. That's all. So, and coming to this part, these cells over the acini, these cells over the acini is called as the myoepithelial cells. Where is myoepithelial? Yeah, myoepithelial cells. Look at it. Myoepithelial cells. Okay, so these cells helps in squeezing the acini. So squeezing helps in propagation of the secretion of the acini. Okay. Myoepithelial cells are present over the acini and not over the ducts. Remember that, not over the duct, they are present over the acini. Striated mucus, intercalated, flat basal nucleus, connective tissue, interlobular duct, and then intercalated. Coming to mixed salivary glands, the last slide of your general histology slides. It contains mucus acini as well as serous acini. Okay, and then again over the mucus acini it contains serous demilunes. Let us repeat again. It contains serous acini, it contains mucus acini, and then again on the mucus acini it also contains serous demilunes. Okay, serous acini, mucus acini separately, and then this thing, serous demilunes over mucus acini. Again, interlobular ducts and intralobular ducts classified into striated and intercalated. Example, submandibular. See, this is connective tissue septa. Now, this is a mucus acini. Okay, mucus acini, and then striated duct, intercalated duct, serous acini, individual. Okay, individually this is also individual. Now, serous demilunes over the mucus acini. See, you can see the mucus acini, right? The flat basal nucleus and a large lumen. Over that we have cells. This is called as the serous demilunes. If at all it is a single cell that caps over the acini, it is called as myoepithelial cells. But if it is multiple, it is serous demilunes. Serous demilunes. Okay, connective tissue, mucus acini, flat basal, interlobular duct. Okay, I hope and that's all, guys. I hope everything is clear. If at all you don't understand some concept or you feel like forgetting, I would suggest you to go through the video again, play it in 2x or 1.x, whatever suits you. Do it, get perfect in this general histology through this video. I have referred Balakrishnan Shetty for the diagrams, and then for the microscopic view I have taken my own images from my lab. Okay, so yes, that's all the best for your exams. Keep grinding, guys. For the next video I'll be posting systemic histology, the complete video like this. That's all. Hit a thumbs up, subscribe to my channel.